Okay guys, so what I'm going to do is I'm going to go through the lecture that we just had today and just re-explain everything just in case you had any issues with it. So the whole point what we were talking about before in the last class was about transcription. So that's the process of going from DNA to mRNA. And now we've got this mature mRNA that has been synthesized and it's going to create proteins, and proteins are what make you you. They are why certain functions happen in your body. They make up everything that is you. And so we want to know how we can go from this mRNA, which is a copy of your DNA, into this protein. All right, so just a couple of terms to make sure we're on the same page. If you look right here, this is one monomer, and this is an amino acid. And we reviewed this, I think, in Chapter 2, really early on in the year. Just remembering the fact that an amino acid is made up of an amino group, a carboxyl group, and then they always have their R group. So this is what one single amino acid looks like. Now, if I take two amino acids and combine them together, and I combine them with what's called a peptide bond. So when I take two amino acids, combine them together with a peptide bond, if I have a long chain of those, that's actually called a polypeptide, and poly meaning many. So you have many peptide bonds, so it's called a polypeptide. All right, and there are about 20 different types of amino acids that can be linked together in order to create a polypeptide. And which amino acid gets placed in the long chain, the order in which they're placed, that's what determines the certain properties of proteins. Okay, so that's just like a little bit of a background about proteins themselves. So you have a bunch of amino acids linked together with peptide bonds that makes a polypeptide. That will eventually, that becomes a protein. Okay. All right, so what I'm going to do right now is go back and talk about mRNA and show you how we go from mRNA to these amino acids and therefore polypeptides, because that's the whole process of translation, which we're going to get into. All right, so the genetic code. The genetic code, guys, is just the order in which you see the bases. So the genetic code, it's just like reading a book. It's a certain language that tells your body, that tells your cells what proteins to make, and we just call that the genetic code. Okay? It's a language that is going to be read, and I'll show you how it's read in a second. Now, the order in which it's read, it's done in pairs of 3. Okay, so it's 3 bases at a time that are read. Okay, and so these 3 letters, what you would consider in our terms to be a word, a 3-letter word, that's what we call a codon. So each of these 3, that's a codon, that's a codon, that's a codon. All right, and each codon, as you'll see in a little bit, refers to a very specific amino acid. All right, now the analogy that I came up with, each base represents a letter, right? If I'm using an analogy. So a base is a letter, which is really obvious. Then a codon represents a word. So if you put 3 letters together, that's a word. What I would say is based on the word, okay, you get a certain amino acid. So based on the codon, you get a certain amino acid. The analogy that I like to use is a part of speech. So if a word is a noun versus a verb versus an adjective, the amino acid is the part of speech. And if you have a long line of different parts of speech, that's your grammar, right? So subject, verb, adjective, adverb, verb, subject, a whole line of amino acids, a whole line of parts of speech, that gives you grammar. Okay? So if that analogy works for you, awesome. If it doesn't, sorry. All right, so let's see how we actually read the genetic code, how we read these codons, how we use these codons in order to produce amino acids. So, since there are 4 different letters, okay, and there are 3 in a codon, 3 letters in a codon, you end up getting 64 different possible codons. So it could be GCA, GCG, GC, etc., etc. There's 64 different combinations. Now, many codons produce the same amino acid. Now, to determine what codon, what the 3-letter pattern, what amino acid that relates to, all you have to do is follow this chart. It's super easy. Okay, so what you're going to do is you start at the center. So let's say my codon is GAA. Okay, so I start at the center. Here's G. Then I find A, and then I go to A here. And so the amino acid that's going to be produced is glutamic acid. Okay, so let me do another one. My codon is CUA. Okay, so I start at the center, C, then I go to U, then I go to A, and that's leucine. So when I said many different codons make the same amino acid, so for example, you have CUU makes leucine, but CUC also makes leucine, but CUA also makes leucine, but CUG. So as you can see, although there's 64 different combinations, they're not 64 different amino acids. As I said, there are only 20. So all you have to do is follow this chart, and you'll always have it, and you'll know what that codon is coding for, what amino acid is going to go with that codon, all right? The other interesting thing to note, although you have all these different names of amino acids, if you notice right here, there's a stop codon. So if I get the codon UGA, what that tells me is that the synthesis of the amino acids is going to stop, and the amino acid, the chain, the polypeptide chain, is going to be released. Okay, so that tells me to stop. There's also a start codon, and I can't seem to see it right now, but there's also a start codon. Okay? All right. So let's actually get into the process of translation and talk about that. All right. So the most important thing for you to know, ribosomes are what are actually going to be reading this mRNA. Now, this mRNA is produced during transcription, which happens in the nucleus. So all the components of transcription that we've talked about so far are happening in the nucleus. Okay, once the mRNA leaves the nucleus and enters the cytoplasm, that's when the ribosome is going to come on in and attach to it. All right, and I'm going to show you and explain it, and then I'm going to show you a video and explain it what happens. So the first step, guys, mRNA just leaves the nucleus, right, and a ribosome attaches to it. Now, let's see what that actually looks like. So here's your ribosome right here. Remember, you've got two subunits. You've got the large subunit right there and the small subunit right there. Okay. Now, as the mRNA passes through the ribosome, okay, a tRNA, which are represented by these guys, the tRNA is going to bring an amino acid, and it's going to bring the correct amino acid, and this is how it does it. If you notice, the tRNA itself has 3 bases attached to it. Okay, these 3 bases we call the anticodon, because these 3 bases are going to line up, are going to find, for example, here's my codon, AUG, so the only tRNA that can be brought is one that has UAC, because they are complementary to the mRNA. All right, so the anticodon is complementary to the mRNA, and that's how it decides which amino acid it needs to bring. So the methionine has the UAC, and since this is AUG and it's complementary to that, that's how the tRNA knows to bring that specific amino acid. Okay, so this just continues on, and you keep adding more and more amino acids. Now, how you actually make the polypeptide chain. Let me show you that. Hold on one sec. We're just right there. Okay, so as the tRNA is bringing the amino acids, okay, so notice we've got 3 different binding sites. Okay, so I've got one here, I've got one here, and one here. Okay, so what you should see, let's first look. Imagine this guy is all the way back here. Okay, so it's got an amino acid, then it's going to move to the second one, okay, and then another tRNA comes in. Okay, what happens between the first and the second binding site, the amino acids are going to bind together, and they bind together by creating a peptide bond. So you can see the peptide bond between the green and the blue circle right there. All right, then it keeps moving through, and so the first tRNA that we had moves to the third binding site. Okay, now what happens is that at that second binding site, okay, once it's at the second binding site, this blue amino acid is going to connect to that red one, and therefore the tRNA loses its amino acid. And so what ends up happening is it moves to the third site. It goes one, then the second, where it loses it to the first one, right, with a peptide bond. Then it moves to the third site, and at that third site, it's then going to be released. So the tRNA breaks away. Okay. So the tRNA leaves the ribosome, and this just keeps continuing. So this one will give its blue to the red. Okay, it's going to move to the third site, and it's going to move off. This purple one's going to come in. This red one's going to give it. So you'll have a green, blue, red, purple line of amino acids or a polypeptide chain, and this one's going to go off after it goes to the third binding site. All right, this continues, and you keep having a polypeptide chain being formed until there's a codon in the mRNA that is a stop codon, and that causes the polypeptide chain to be released. All right, now a polypeptide chain is not a protein. What has to happen is that the polypeptide chain has to curl around and twist, and it's going to make a three-dimensional shape. That three-dimensional shape is what we actually consider to be a protein. Okay? Now, I think I've just got one more. Yep, we were just right there. So this is the last one. The central dogma of biology. I can now say it because we've gone through this whole process. The central dogma of biology states that you go from DNA to RNA, then to proteins. And this is a quote that I found in your textbook that's really important. It says, proteins are microscopic tools, each specifically designed to build or operate a component of a living cell. So proteins, even though we always consider our traits to have to do with DNA, DNA is the master plan, RNA is the blueprint. Protein is what we actually see. When we see hair color, when we see skin color, when we see whatever, intelligence level, everything, that's because of enzymes, it's because of proteins that are functioning in your cells. Okay? So I really like that quote. Now, I'm going to show you this video really quickly, which goes over all of the different steps. Okay, and it's just from YouTube, but I really like it. I'm actually going to put it on mute because I want to explain this as it goes. So if you notice, this is the mRNA, all right? This is the small ribosome subunit, okay? And so at this point, they call this the initiation site. This is just your start codon, so it tells your ribosome to start translating the mRNA into protein, okay? And so that's there. You've got a tRNA. Okay, so here's your tRNA, and if you notice, here is your amino acid. The red is your amino acid. Okay, this is your anticodon, so this is going to match up with the mRNA. So notice A with U, A with U, C with G. So that tells you which tRNA you need. Okay, so that's the matching up, and it's actually going to show it getting going. Okay, so this is just one type of amino acid, which it's showing you, which is really nice. Okay, now, it stopped. Hold on one sec. I'm going to have to refresh this really quickly. Hold on one sec, guys. Sorry about that. I might not be able to go. Okay, so I'm going to stop this video right now, and I'll create one more video to explain this with me doing it verbally. So yeah, there's an error. So stop this video and go to the next one, please.